But the confessions, I mean, the first one is kind of occurred to me about a month ago, which is, by organizing the meeting in Guelph and having all bio and my students and others participating, I realized that I have nothing to say because they're going to be presenting all the work. So that's the sort of first challenge I faced, and I'm going to try to sort of deal with that. The second one is, like inviting the previous two speakers. That's another confession. I wish I had not done that. So again, the work that they have done, and like they kind of both been sort of, mentor and for my work, and I've learned quite a lot from them. So bear with me as I start to sort of discuss some of the work that we have done. Now, I tried to kind of find kind of beautiful maps and things, and this is what I kind of stole from a talk that Donald Bird, my co-conspirator and colleague in Environment Canada, showed, and it's from a World Bank visualization. It's about remoteness and connectedness. And so I think, for me, as I kind of start working on more remote sites, where there is biodiversity, or what we think there is biodiversity left, and we saw from Ted Yahara's talk, for example, the Asian perspective of the forests, I mean, these are sort of basically a good map for us to sort of think about how we're going to build up capacity to move towards what Paul envisioned and Mark Blackster just kind of put in the stage of a framework of what we can do. So, and what I think that we need to think about is that how the DNA barcodes and the information that we get, which reflects to biodiversity, can be linked to all of these socioeconomic and societal and scientific endeavors that are kind of linked to where biodiversity resides. And in our nation, this is a huge issue. And again, it's not very difficult to find out, places that are quite remote and not populated, but they have been exceedingly, under the, impact of, resource development. And again, this is a catch-22 because our economy is based on that, so we can't just move away from some of these, without thinking about the consequences. And as I move and look at the media very recently, this is from, not long ago, what is happening, in very remote areas where I think there are kind of frontiers for discovering biodiversity, and so the impact of those things. In these cases, I kind of try to categorize the type of challenges or the type of things that we need to do, and I will be short and quick because other speakers touched on those concepts. In many cases, we need to track species that we know are at risk, and this is an example of marine mammals, and they're charismatic. They're also been studied intensively, and they're linked to the ecosystem health, and that's why and unfortunately their populations are decreasing, and in many cases. So in this case, we do need tools and to go in and identify and track them. Sometimes, a species level is sufficient, but we may need to get to the population and individual level. I've learned, for example, the, eastern gray whale, that's so rare that, we really need to kind of track the individuals of the tribes. And another layer of dealing with this is to map their habitats, and that's another sort of layer of information that's going to give us the dimension of the biodiversity data that we need. The food that they consume, or the whole ecosystem that enables these organisms to live in an environment. Another, I would say, angle that gets into kind of more ecological work, and I give a practical, again, application or practical sort of real-world scenario, is the natural resource management. And I've been working with the scientists and managers of our own agency here, and they are sort of developing, and they have been developing programs, and they do need more information, biodiversity information, to measure ecosystem services. And in this case, the health of the forests, as is reflected in the health of the soil, the health of the rivers, and the streams that pass through them need to be considered. And another sort of similar, but like with kind of a different sort of scale is to track the cumulative effects in ecosystems. And I give you an example, sort of north of where we are, kind of far north in northern Ontario, we have this system called Ring of Fire. And as you probably know, northern Ontario is quite famous for mining activities. And the Ring of Fire is just becoming feasible, and the licenses have been issued for mining. And the government is thinking, like how can we kind of plan by developing tools based on DNA and other technologies to track the cumulative effects that may come as a consequence of the development of these. Melania did a great job talking about invasive species. And again, like Paul is in the audience, so I mean, and he has done a lot of work on this and others. But the example of, like these are the list of, like species in our own neighborhood, in the Great Lakes that have been invaders, and they've done the damage, they're doing the damage. And then there are some cases that, like we are barely kind of holding, with the killer shrimp or Asian carp, and you've seen in the news. So how can we basically utilize these technologies? So DNA-based technologies to deal with them, and you've heard. Now, I'm not going to go into more details. Just going to show you a photo of Paul. No, that's not Paul. That's Fred Sanger, and that's how it started. And in terms of like DNA barcoding, and I'm saddened by the fact that Sanger died. And but when you look at the legacy that's been kind of generated, the knowledge, all of these things is based on the technology and the approach that was developed by Sanger, and then now we can see many other methods that are being developed. But I mean, I made this cartoon in 2007 thinking about how biosystematics from population to phylogenies can utilize DNA-based information. And the reason I did this over Christmas with Donald Hickey and Paul and Greg Singer was to try to position DNA barcoding within the context of biosystematics in terms of sampling, in terms of sequencing, and in terms of the type of information we get. And that was mainly, a way to try to articulate the mission of Barcode of Life and, capturing all the DNA extracts and then using them for doing deep phylogenies and adding more genes here, or adding, cases that we are kind of finding interesting patterns. And I've done this with Dan Jansen, for a number of cases, and it warned me not to get too distracted in terms of understanding, the local, adaptation, cryptic species, population level variation. So I actually think that in 2015, with the new technologies and how barcoding has evolved, at least I'm going to revisit the way these arrows are pointed, or their kind of size and how they're going to work. And perhaps like we can see that this becomes more like one unified system, where we can look at a barcode as an entry point, and then like quickly generate a lot of more data. And I know Eric Kuzak is going to talk about some of this tools based on shotgun sequencing and so on. Now, fundamentally, I think we also need to ask this question as we go forward. Mickey put this in the context of ancient DNA. Is really we are at the stage that we can engineer genomics tools and information systems to address questions. It used to be that we have Sanger reads that are this much, and we have primers that are like good for this group and that group. I think that and again several years ago we looked at this from a barcode size when we were struggling to deal with museum samples, and we were thinking about adopting and using next-gen sequencing technologies. Is that we did this sliding window analysis where we basically, chopped the barcode sequence systematically, and then looked at the proportion of the species that are detected, and we kind of converged around 300 base. So essentially, the current barcode length to me is an even overkill, but it's required because when you think about the sequencing technologies and the error rate, I think that we do need to keep the barcode standard as it is, at least for animals, because again, we need to have some more confidence in the data that we are building. And as it's been shown, the primers are working well, at least for many groups that we have looked at. So moving quickly towards whole genome approaches, I think that needs to be taken, with a bit of caution when you think about the mission and when you think about the level that we are looking. How do we obtain sequences? So, basically, as you all know, or gold standard preferred and enabler technology, in this case, been Sanger sequencing and the approach that's been used in collection-based research, sorting organisms, identifying them as much as possible, as fast as possible, and then moving towards sequencing. And you can build a very high confidence sequence library using this. And I grew up with this technology from my graduate work, and I still send some of my sequences to Sanger sequencing. But I think that we also have been cautious, have been careful to kind of think about the possibilities with next-gen sequencing, and we have seen many examples. And Shadi Shakrolla in my lab developed this fantastic approach of working with this sorted material with next-gen sequencing. The workflow still got some limitation in terms of bioinformatics and also streamlining and provisioning specimens, but it's coming, and I think that it's quite time now to think about this a bit more seriously, and people who have the possibility to do comparisons, build informatics tools, just as we were developing the barcoding, and it took about 20 years of Sanger sequencing to the point that we started utilizing it for barcoding in a serious way. I think that we are like 7, 8 years in the next-gen sequencing age, and I think that we are seeing some of these type of technology. The good thing here is that you get a lot of coverage, so you can, and it's clonal and it's parallel sequencing, so you can see if there is a Wolbachia or if there is a pseudogene and those things. But again, it's not been matured. There have been a couple of papers, at least on the barcoding side. I know that people have been using this in genotyping and medical side for quite a while. But I think by and large, as you have heard, we are using next-gen sequencing for bulk specimens, and I'm not going to go through that. Most of my talk is going to focus on what we have been able to do and what is maybe possible and what I don't think is possible at this point. So this kind of idea of doing sequencing from bulk or eDNA kind of have been named with various terms. Some of them, I think that, are more imaginative in terms of opening the barcoding, the meta-barcoding, so moving away from just a standard barcode. And some of them have been kind of rooted to genetics work and so on. I kind of use this term because I want to have everything here. And again, I think that the information we get allows us to go from in various levels of organizations. Here, this bubble chart is an old one, but it's still quite valid in my opinion, given that we have the libraries of these genes, and we have taxonomic resolution, for various groups of organisms. And as you can see, most of these are actual barcodes. 16S is kind of the de facto barcode for what we call bacterial species, even though bacteria don't kind of fit into the species so much. And then 18S is a pre-barcode for protists, and been used also exceedingly. Mark, I think most of this is from the nematodes because this is the soil data. And then, CO1, ITS, and RBCL are all designated DNA barcodes. And I throw in the 28S because it's also been used quite a lot for fungi and other organisms. So I think with this handful of genes, perhaps, I think that we can solve this biosystematics sort of puzzle from the community-based data. And I think, on an individual basis, we can add more nuclear genes and so on. And I think that we have started sort of, doing some proof of principle around the environmental applications. And this was a paper that came out quite a while ago on bentos with Donald Bird and the team of Environment Canada. And Donald wrote this perspective article, how this information can move into a real-world biomonitoring approach, this notion of biomonitoring 2.0. I helped Pierre Tabernet, Eric Kuzak, and Lauren Riesberg to kind of edit this issue of molecular ecology that it's kind of aging now from 2012. However, it has a kind of a group of various studies and perspectives and technical reviews on the use of these approaches, in various shapes or forms. And technically, we thought about another layer, which is, we've heard about Malaysia Straps, and, I live in a space that there is a lot of Malaysia Straps around me, like in bio and everywhere and all the backyards around us. So I need to think about it, how are we going to use it? I mean, as me, who is not so much, into organismal biology, I thought, okay, can we get all of them in one shot, but also microbes that are there? But that's kind of opened up a new interface to biodiversity data. As Naomi mentioned in her talk, we are capturing the arthropod data, but we are also capturing their bacteria and fungi, but it's not linked to individuals. How can we utilize this information? How can we, put these in a perspective of biodiversity assessment and environmental change scenarios? But doing this, we also, just like Mickey, we like to kind of play a little bit in the technology space, and this was an attempt to sort of look at the number of primer sets that are needed to kind of get to the point that you can say, I've got everything that I can. And in this case, we looked at combinations of the primer sets for just the CO1, part of the CO1 barcode region, about 350 base pair. And as you can see, there is value in adding more primer sets, but it's kind of, getting to the point that it plateaus. And right now, we are routinely using 2 or 3 primer sets for our mini barcodes of CO1, and I mean, in some cases, they sit on the same part of the gene. In some cases, we do the front with 2, and then we do the back end with another 2. But we haven't stepped out of the barcode marker for animals, because we do want to take advantage of the library that is growing. And I encourage all of the people who feel that primer is an issue for at least some of the animals that we know that primers work in individual specimens, reach out to the community. And this was what Karen James mentioned in Next-Gen workshop we had. And I saw the tweet by David Schindel saying that, why are the metabarcoding people are using non-standard, markers, as we are building these libraries? This is an idea of, we shouldn't, as ecologists or molecular ecologists, separate ourselves from taxonomists. That separation can be dangerous for the field. For many years, as I've learned from Dan, ecologists have relied on taxonomists. And taxonomists are using DNA barcodes in a standard fashion, and they're building these libraries. They're finding fantastic patterns and describing diversity. And as ecologists, we are moving away from using that because technically we feel it's impossible to sort of use a standard marker. So don't want to sound like a priest here, so I'm just going to move on. So the other sort of thing that we have learned is, as we combine, get this data from metabarcodes or environmental barcodes and combine them, we get a lot more information from a system. And this is a Malaysia Strap again. This is a published work, and so I'm not going to get too much into it, but we always see this value of adding. And some of this might be really false positives, sequence error, but some of it's like layers of diversity that we can't capture in individual sequences. Now, I started thinking, as I went and gave a talk in McGill, and Graham Bell asked me, oh, have you started thinking systematically, asking ecological questions? And I have a group of friends here in our department that are familiar with ecological questions, and we have started thinking about what we can do with this data in an ecological environmental context, and also thinking always about the applications and how it can be used by agencies. That's been my focus. Now, but again, I went to Dan to begin with, and I said, can we, the Malaysia Strap we did, with Joel Gibson, can we get a bit more? I sent 9 Malaysia Straps with a student, and Lisa Ledger is going to talk about this work in the conference. But we started thinking about a system that was there and very familiar by Dan in terms of looking at this area is really sitting in the dry forest side, and there is not much any variation other than the fact that one is a primary forest patch, the other one is a 70-year-old succession, and then there is this fire break that they have kept there. And so we asked very simple questions, can we measure beta diversity using this approach? And in this case, we got the sequences, and we used the actual library to identify the sequences at the genus level, and it holds. Even though there is quite a bit of variation, we can see that the differences are clear here in a statistical sense. And Lisa did a whole thesis on this, and it's coming along, and she's going to talk about it. It holds well with OTUs and even better. But even if you go with the identified organisms, you can see. And what are these organisms? Obviously, we have focused on arthropods, and you can see that in these sites, you can see the distribution of these different groups of arthropods, and some of them were a little bit sort of unusual for us, but I think that's kind of, we can imagine how they can get in there. So, and now I think that this type of work can help, hopefully, what's happening down in the middle of this remote, to be, like not literally, but forest in Costa Rica, which is this geothermal, activity. The country relies on these type of, tools to foster their economy, but it's in the middle of this protected area, and can we use these tools to deal with the, like assessing the environment? And the good news is that, like these are being actually proactively pursued by some of these industries, if it's pitched correctly by someone like Dan, obviously. So, and that's what's happening right now. So, in here in Canada, and going up north a little bit, and not necessarily the Arctic, but the boreal forest, and we started this project with the help of our agencies, Environment Canada and Parks Canada, and then basically this is where the area is the Peace Athabasca Delta, and the Alberta oil sands are near, and also hydroelectric activities. And it's a very valuable ecosystem, and it's been named as an ecosystem under sort of at risk and under threat, and it's obvious why. And I think we started sampling intensively to do the meta-barcoding, environmental barcoding, with the help of our agencies. And I'm just going to glance at some of the latest kind of findings that we've had with this. We're still crunching the data. And here, for example, I'm going to show you how we sampled most of our sites with repeats, and in some cases with soil, we had a lot more repeats. And then the data that is coming out holding quite nicely. This is the OTU level of benthic biodiversity. All the triplicates of the sites stick together in just unsupervised clustering algorithm, and that's encouraging. But we needed more than that. We needed to compare this with morphology, because that's the standard for biomonitoring with aquatic systems. And this is the work that Joel Gibson and others have done, and kind of idea of comparing biomonitoring 1 and biomonitoring 2.0. And so here I'm going to show you a little bit of data. Joel is going to talk about it a lot more tomorrow. In this case, we are looking at these two parts of the Peace Athabasca Delta, these two rivers. And as you can see here, very nicely we can separate the gamma diversity at different levels. With the morphology, we have the data here, the DNA at the order level, family level, genera, and a species, and and it holds well. Also, you can do the OTUs and separate the OTUs, as I showed you earlier in the graph. Now, I'm going to pause a little bit and ask this question. Why do we need the eDNA in some cases? And I think that's part that I kind of want to go back and sort of think about some of the stuff that I'm doing. We've heard that okay, in this case there is this animal in the water. Do we need to get the water and get the DNA of the moose? We can see the moose. So in many cases we have tried to kind of get into the sort of extreme. And in the case of bentos, I think that when we have kick nets, and other methods to get these bugs out locally, going after the water might not be. So I asked this question, we have the data from wood buffalo. We sampled water and bentos in the same site. And so we said, let's ask the question, can we separate these, in here we have water and the bentos in the same site. So I'm just going to show the data for one site. So comparison of the eDNA in water and bentos. So if we wanted to see that, water can replace the bentos, so we bypass getting the sample from bentos, then obviously we want to see a pattern of, water, bentos, water, bentos, water, bentos in ABC. However, that wasn't the case. So here what you see is that the benthic samples kind of cluster with each other, and the water samples cluster with each other. So that shows that, like at least in this method that we have used, like there is not the same kind of biodiversity reflected from benthic to the water. This is a small system, and this is very preliminary. But when you look at the data, by and large, again, you see a drop of about 50%, qualitatively, and there are things in the water that are not in the benthos. So I think that's the sort of thing that we need to be cautious about. I have about 7 minutes left, so I'm just going to go a bit faster. We have a lot of good tools to look at vegetation and the surface, using satellite imagery, radars, and so on. But when it gets to the below ground, we really don't have much ability. And so we thought in wood buffalo, again, these are highly dynamic systems. Nicole Fauner told you this story, so I'm not going to go in detail. I'm just going to show some complementary data that we got from the soil data for plant diversity. And here, again, going into tweaking the sort of marker space and, what can be done with multiple markers, we can clearly see that if we combine these plant markers, and 3 of them are a standard, or 2 are a standard, and then 1 is kind of additional here. And then we added 1 that is also being used, a lot for diet and for soil analysis. We see this idea of the, like complementarity. And as Nicole mentioned, like we need to think, like about the libraries carefully when we deploy these markers. This is what I like more because it puts in the phylogenetic perspective. So we got the tree of life of these groups and then fitted the data that we got into it. And as you can see, some groups are just illuminated, lower plants and so on, like with some markers. So I think this idea of multi-marker, as Mickey mentioned, in some cases is very important. And the length of the marker, as again, was mentioned, for contemporary data, we need to think about longer markers because then we're going to have ubiquitous, like data from the soil at least. And this is, again, reflected in the work that Nicole Fauner has done in my lab. And then the DNA marker complementarity is going to have another dimension when we are looking at, like association of different organisms. In this case, we're looking at plants and fungi in soil. And I'm kind of certain, and it's already been shown in a number of publications, this type of associations using DNA barcode data, especially in the meta-barcoding framework, is going to be very powerful to give us understanding of systems that we had absolutely no ability to look at. These are below ground, sequences, and again, as you can see, like we can tease apart fungi with these 2 markers that are kind of geared towards it. And there is going to be a talk, again, on this by Terry Porter, and she's on mat leave, so she's going to come tomorrow just to give her talk. And I'm not going to go into it. Now, in terms of the analysis, I think this is what we have seen, throughout the meeting, and especially for meta-barcoding. And I think that this is something that down the road we need to think more about it. The norm is we get the sequence data, we do our taxonomic assignments, and then we go and do ecological analysis. That's been pretty much the norm, but the meta-barcoding is shifting this paradigm. And I think in this case, you get the sequence data, you have your OTUs or bins or haplotypes or whatever you want to call them, and then put it in a matrix, like use a program like Vegan or something, and get your basic ecological analysis done, and then tag your sequences with what they are. This way, what we have done and what we have seen in our data is that if you go with this route, as Mark also showed, you lose about 70–80% of your data, depending on where you work. And so the power of the ecological analysis will be reduced quite a lot, especially if you're looking at short changes in the community structure and so on. But we don't want to, like give up all of the information that is out there. So once you're done, then you can tag your sequences using the best approach that is available in the library so that you can get more biological and in fact, test some of the hypotheses. So I think this is going to become like that. So you go back and forth here in terms of additional information. Another, I would say, consideration that I want to add here rather quickly is in terms of sampling, both in terms of sequences and in terms of actual environmental samples. The sequences are getting very cheap. The read lengths are good. And I think that it's time now to go into kind of sort of high-frequency sampling of the environment. Not just for microbes, it's been done, but I'm thinking more about like larger organisms. Like in terms of, this may allow us to actually move with the repeated sampling into identifying the rare targets more robustly, but possibly developing tools and protocols for quantitative analysis in terms of transforming the presence or absence data into the quantitative analysis. This is what epidemiologists have been doing for decades, I think, and ecologists have also worked on it with occupancy models and so on. It just needs to be, demonstrated, and I think we need data for it. That's data temporally and spatially. And so I think, these are the case studies that, like we have done, and I'm not going to talk about, that's my confession here, basically, that I can't get into them, but there are a number of these talks that, like throughout the conference, and one poster that are going to talk about various ways that we have played with this. There are collaborators from Portugal, France, and other places that, like they have done quite a lot of work. I'm excited with what is happening in Norway, Netherlands, and so on, Germany. And I think, what we thought, a number, as I got started working on wetlands, I need to, again, learn about wetlands and learn about, and Braulio mentioned, how important wetlands are. So we thought about like putting in the context of the wetlands. Wetlands are interesting because of their value, but also interesting because of the habitats they harbor, and because of the, like also the limited number of them, unfortunately perhaps. But we are starting to think about like, like joining forces with Ramsar, the UN Convention on Wetlands, and trying to sort of pitch, like this type of approaches to build, like a next generation wetland biodiversity and functional assessment, system and deploy that. And in this case, like we can, I'm going to glance at it and then quickly finish. We can assess biodiversity. We can also get into functional analysis with some of the transcriptomics work. You can look at the functional genes in two ways, adaptations, and what's happening in terms of rates of evolution of these genes. But there's another layer, which is very short-term, changes at the individual level. This is the work that mainly ecotoxicologists have been doing. And I know that Paul warned me, like not getting into that because it's going to get complicated. But I actually feel that we can define functional barcodes for some of these, like systems and deal with the issues that they are facing. Just like the way barcoding has started helping biologists, in the taxonomic context. Right now, we have very rare cases of comparisons between methods and species when it comes to these changes in the environment. Few methods are available to look at sublethal. We have to wait for organisms to die practically in the lab. And then limitations in terms of ecological effects, because the work is done in the lab mainly. And then we can't build a causality, like with what's happening in terms of the phenotypic changes. So this is the system. To me, this is a very much barcoding system, really. This is the, like the space that we have with all the community level, and then the stressor and where they react. And I think that the transcriptome level will give us this data. And if it's metatranscriptome or targeted metatranscriptome or functional barcodes, it can give us quite a lot of data. In Kunming, I introduced this idea. I'm going to just very quickly, in 1 minute, show you some data. The data comes from the Great Lakes sediments, especially the PCBs that have been very nasty chemicals deployed in these sediments. And then we started using a bioinformatics pipeline to identify targets. You can do a transcriptome, full transcriptome scan to identify your targets, but we went with a shortcut, and this is the work of Nicole Fauner, Gina Caprezza, and in my lab, and then basically identifying these targets using this algorithm, moving them into the organisms that are now used because we want to have some comparisons. In this case, we are using hexagenia, and then exposing the hexagenia in the lab with controls. Interestingly, we started about 200 targets. Once we developed our probes and the generate primers and so on, and got our MySeq data, and then did the experiments with the exposure, we see these are the three genes that are quite relevant. There's quite a lot of literature about them. But interestingly, there hasn't been any work in the invertebrate world on the PCBs. Most of the data comes from vertebrates. And now we are investigating what pathways these genes are going to be impacting. But we do see a significant change in the expression of these kind of functional barcodes, if you will. Sorry, I went a couple of minutes over time. That was my final slide. Thanks again for your attention and for being here with us. And I would like to thank my lab for doing all the work, the organizers in bio and across the world and helping us, and also the funding agencies. And thank you very much.